It's my pleasure to moderate the coming session featuring two outstanding early career neuroscientists, Audrey Fan and Sergei Stavitsky. Both Audrey and Sergei were Tsai interdisciplinary neuroscience scholars during their postdoctoral training here at Stanford. My name is Miriam Goodman, and I have had the honor of directing and co-directing our postdoctoral training programs here at the Tsai Neurosciences Institutes over the past decade. Our postdocs bring their expertise, creativity, ingenuity, and ambition to our research community. In their honor, I would like to invite postdocs in the audience to ask the first question of our speakers when we get there. So let's get started. Our first speaker is Dr. Audrey Fan. She is Assistant Professor of Biomedical Engineering at UC Davis. As a postdoc at Stanford, she was a member of our very first cohort of interdisciplinary scholars. And as a postdoc, she worked to establish simultaneous PET and fMRI imaging to generate improved measures of blood flow. Today, she holds a K99/R00's Pathway to Independence Award from NIH and runs a vibrant research group and co-leads the imaging core at the Alzheimer's Disease Research Center at UC Davis. I'm really looking forward to hearing from Audrey and to knowing what she's been up to since her time here at Stanford. And I encourage you to welcome her to the stage.
Thank you so much, Miriam, and I'm just completely honored and excited to be back here today and be able to share some of our imaging results to study brain physiology. As Miriam already described, I want to share a little bit about myself. I actually did my undergrad here at the farm in electrical engineering, where I really fell in love with signal processing. I took that to MIT, where I also received my PhD in electrical engineering and dove into MRI physics and image reconstruction. But it was really as part of this inaugural cohort of the Tsai interdisciplinary postdoc program that I felt like I became a neuroscientist. And I have the community here and also my fantastic mentors, especially Greg Harchuk and Gary Steinberg, who you'll notice are both clinician scientists. And so this really helped me to understand a clinical and neuroscience need that I could apply my engineering skills to. And at the time that I was starting my postdoc, they were just installing the first GE PET-MR system, a hybrid system where you could simultaneously measure PET and MR. And I'm going to start a little bit by describing my postdoc project that was funded by the Tsai Institute. This is motivated by a physiological and clinical need where our brain is only 2 to 5% of our body mass, but it's incredibly energetically demanding. And therefore, to meet this incredible oxygen need, we constantly need blood flow delivery through arteries to the brain. And in participants, patients with diseases such as moyamoya disease, what you'll see is that arteries delivering this blood supply to the brain become occluded. And this may lead to twisty, tortuous vessels, structural changes in the brain vasculature that you can see here. But potentially even more important is characterizing what this does to the perfusion, the actual blood flow patterns, because some patients may be able to compensate with collaterals and others may not. So what I dove into during my postdoc was learning about these imaging methods to serve patients. And in this context, O15 water PET, so positron emission tomography, literally with water containing a small amount of radiation, allowing you to generate a blood flow map of the brain. And in this participant, we have a unilateral disease, so you see reduced blood flow on what appears on your left, which is actually their right hemisphere. And my task was using the simultaneous platform to advance, optimize a non-invasive, non-radioactive MR approach called arterial spin labeling. And I was able to do this with appropriate timing so I could capture blood flow maps non-invasively without any injections to help these patients get a perfusion assessment. What was inspiring to me, and will kind of be a through line through the rest of the talk, is that we weren't interested just in baseline, but we designed a brain stress test. So similar to how a cardiologist might have you run on a treadmill, we developed a paradigm where we used a vasoactive stimulus. So kind of like an fMRI where you might have a participant perform a task, we gave a drug. Sometimes this is done by breathing elevated levels of CO2, but we gave a drug called acetazolamide to dilate the vessels. And this response, this increase in perfusion, can be detected by BOLD, which many of you are familiar from fMRI, but for my project, especially with perfusion. And this is what the map looks like after vasodilation. And what's really interesting is in this patient, there's actually paradoxical decrease in perfusion, meaning maximal vasodilation, and a loss of autoregulation. This is a participant that really could benefit from endovascular intervention. And we were able to recapitulate it, again, with the non-invasive MRI approach. And so to me, it was really interesting that not only can we measure physiology, not just brain structure with imaging, but that we could look at different dynamic physiological states that we had control over. And through my time at Stanford as well, by virtue of scanning at the PET-MR, I met many researchers who were also interested in related disorders. And I say related because there's now a broad swath of neurodegenerative and neurological conditions where we know, for instance, Alzheimer's disease has a parallel but also intersecting progression of neuropathology. And this is really inspired by Beth Mormino, who leads the ADRC here at Stanford, and has really inspired me to kind of think about how physiology, more subtle changes and dynamic changes, could be more sensitive to early aging-related or neurodegenerative-related diseases before the onset of structural disease. So with this background, I'm going to share two projects that we've launched in my lab now at Davis around this theme of dynamic neurophysiology, and one alluding to a previous presentation having to do with fingerprints using imaging, and how we've used neuroimaging repositories that are increasingly becoming available to look at brain-body physiological connections. And my hope is that this has broad impact because these approaches are faster, dynamic, and can detect subtle changes that it will be maximally impactful for many neurological conditions, as well as give us some basic science understanding of familiar signals, like resting state fMRI signals that some of you may have heard of. So launching into one project is led by Greg Wheeler. He's a BME grad student in my lab. And his project revolves around designing a method called dynamic vascular MRF, vascular magnetic resonance fingerprinting. So if you've seen a lot of MRI images, we've commonly tout them as having, exquisite contrast and soft tissue sensitivity. But what we're thinking of is looking across time points called echoes in a scan. If you look at the contrast, the signal, as it evolves, you can start to think of the MR signal not just as one static picture, but as a signal curve. You're seeing my signal processing flavor coming in already. But we want to make these curves informative about physiological parameters such as blood volume, tissue oxygen saturation, and vessel radius, exactly along the lines of these physiological, subtle indicators of brain health. So what we've done is to think about this paradigm as fingerprinting. So just as a fingerprint in our day-to-day thoughts of it is that you acquire a fingerprint, the components of the fingerprint allow you to match to an existing database, but it's that database match that then reveals underlying information associated with the match. And similarly, if we now start to think of a single voxel and its curve for a particular sequence, this match, compared to a portfolio of possible curves, will help you tie to the underlying parameter, in this case, volume, radius, or oxygen saturation. And it's really this key simulation platform. So what we're doing is a lot of biophysical modeling of the microvascular in these tissue voxels that allows you to create these curves, each with an associated CBV radius and SO2. And in this case, we're actually, for those of you who are MRI aficionados, probing the mesoscopic regime that are most sensitive to this microvasculature with a relaxation approach called R2 prime. So this is what the sequence in the curve looks like. This is something that I already worked on in Greg Zaharchuk's lab in tandem with another postdoc. But when I was here, our MRF approach was static. It took 4 minutes, and it did work, but only if we injected contrast, and that's not appropriate for dynamic imaging. We needed the contrast to help dissociate certain parameters and gain sensitivity, but now we're trying to get around that, and building off of this approach, our unique approach is to perform dynamic fingerprinting. The ways we're going to do this without diving into the technical details is with a new sequence that we've collaborated to implement that has acceleration in plane and through the slices of the brain so that we can get multiple echoes, 5 echoes, in 5 seconds. And then we also have a better matching approach that uses complex values and also iterative matching so we get around some of the sensitivity issues. All that said, here's a representation of the sequence. We're getting 5 different echoes from which we can extract a curve, and we're doing this every 5 seconds. And so the way I think about this is imaging non-invasively, no contrast, but it's essentially getting you a multiplex neurophysiology. So every 5 seconds, we want to produce a map of CBV SO2 radius. And so some of the steps we've taken in this project first are to, as an engineer, being methodical, retrospectively undersample. And so going from our original 40 echoes to 5 echoes, we've done this in simulation studies. Here is a sensitivity profile where the lighter colors indicate good matches, so certain fingerprints that could be potential candidates between CBV and SO2. It's not perfect, but it is robust as we reduce the number of echoes. And from our retrospective in vivo data in humans, we're also undersampling, creating these matches with our biophysical simulations. And while there is some change to the quantification of SO2, overall, the platform seems to be able to provide robust physiological parameters. Again, every 5 seconds, we're able to get a map like this. And where the project currently stands in our recent results is going back to the idea of doing a vascular perturbation. So now we're having participants breathe a little bit of CO2 as a vascular paradigm in a block format to perform vasodilation of brain vessels. And here are examples of the end-tidal CO2 trace, so what's actually happening in the expired breath in these participants, and the associated SO2 over time that we are measuring with our fingerprints, where then you can also create a map of the response in SO2 quantitatively during this experiment. So we're really excited. This is just one parameter, but again, we're doing this for multiple parameters. And our ongoing study, which I hope to share more with you next time, is doing this exact test in younger versus older adults as a stress test dynamically, because we think this sort of reactivity is a sensitive change that we could detect before the onset of cognitive decline. It does associate with cognitive measures in other studies. And we think even perhaps the timing of the response, not just the magnitude of the response, will be interesting in understanding this healthy aging process in our brain vasculature. So I'm really excited that we have this platform working, and I'm hoping I'll get the chance to share results in our group level analysis in the future. Now, I think as we think about the brain and aging and vascular health, I think it's also important not to consider it in a vacuum. And one way that we've thought about physiology also is to link it to systemic signals, including heart rate and respiration. So I'm going to bridge now to another dynamic project led by my neuroscience PhD student, Quimby. And she's using spectral and temporal correlation analysis to understand how heart rate and respiration affects resting state bold signals. And the idea behind this may be familiar to those who have worked with resting state connectivity. So we have a bold signal in two different brain regions. If they co-fluctuate over time, if they're coherent or have a correlation over time, we think that the brain regions underlying that correlation are functionally connected. But as many of you also may know, the bold signal is not just neuronally driven. It's sensitive to local and systemic physiology, vascular coupling. And because of that, it actually may reflect long-range correlations with systemic physiology, like heart rate and respiration. These effects are often removed as a nuisance during whole brain regression in the pre-processing for resting state. But what if we thought about this as another source of information? So launching into this is also the intriguing finding from previous studies actually done by a colleague at Stanford, where if you think about a bolus of a respiration response or cardiac response, and you characterize this across the brain using, say, k-means clustering, voxels that tend to respond in the same way for bold to respiration or cardiac tend to cluster in networks, and networks that we're already familiar with. And so that's also really interesting and led us to pursue this investigation on a network level. We are aiming to correlate these resting state bold signals with heart rate variability, as well as respiratory variability that you can measure with a pulse ox and a respiratory belt, respectively. And what's also interesting is that each of these heart rate variability and respiratory signals, they have specific frequency fluctuations. So it was important for us when we looked at this association with systemic physiology dynamically and the brain bold signal that we also consider the spectral or frequency components. And we did this leveraging some fantastic new repository data from the Human Connectome Project. We're looking at a subset of 50 participants. And this Human Connectome Project data had concomitant peripheral physiology recordings, which we leveraged to, first of all, extract bold signal from resting state networks, look at the corresponding heartbeat interval and respiratory volume per time. So not the breath to breath or heartbeat from beat to beat, but its change over time. And then we're looking at wavelet coherence, so an advanced signal processing method that looks at synchrony, correlation over time, not only at different time points, but also across frequencies. So let me just give you one example in one person of what this looks like. This is a wavelet coherence plot of heartbeat interval and the visual network functional MRI signal at rest. And along the y-axis is frequency in hertz. And what's really interesting is that there are in the x-axis is time over the duration of the scan. And what's really interesting is that there are these two frequency bands, as we alluded to before, that biologically seem relevant to heartbeat interval, but have strong coherence. So these two signals of heartbeat interval and the visual network, bold signal in the brain, are in sync, but at specific frequencies. And these frequencies are biologically meaningful. And then if you look across time, the x-axis, this is not always the case. So there are time points during the scan that they're more in sync with each other than not, and which is intriguing. Is this indicating different autonomic or vigilant states? I find that aspect of variability really interesting. If you average across the time and you just ask the question, okay, across people and across time points, what is the coherence? Again, you're seeing these two frequency peaks of coherence. And there are certain networks, such as the somatomotor and visual networks, more sensory, and the ventral attention network that tend to have higher coherence with heartbeat interval, especially at this lower frequency band. We did the same with the low target frequency band of interest for respiration. I'm sorry, this should actually say respiration. And again, we see that's ventral attention and some of the sensory networks tend to have more higher correlation. the last piece of analysis with this data that I find the most interesting is actually looking at phase. And phase, from a signal processing perspective, could be, you could describe it as like a lag between the signals, and we often can represent it with like a vector. And so if you overlay in the yellow parts where there's high coherence and you look at the phase, that's telling you which signal is coming first. And what's really fascinating to me, so here is a frequency on the x-axis, coherence on the y-axis. At the lower frequencies where there is coherence, it seems that bold is leading the vascular peripheral signal, so a neurogenic autonomic, perhaps, response, whereas in the coherence at slightly higher frequencies, a vasogenic, maybe the physiology driving the sync within the bold signal in that network, is actually the opposite lag. And we saw this in multiple networks as well. So I think this is fascinating because it helps us understand physiological sources of the bold signal, bidirectional signaling between brain and peripheral physiology. And I think it gives a little context into some of the technical advancements, especially with Human Connectome, where they're really pushing the sampling rate, they're really thinking about functional connectivity at higher frequencies as well. The higher frequency information actually could reveal different signaling patterns, and I think that's fascinating. And there's many reasons to continue this work to look at why these canonical functional networks, whether it's sensory versus cognitive or their anatomical location near vessels, what drives certain networks to be more in sync than the others, and how might this change in, say, mental health disorders that have autonomic dysregulation. I think this is a fascinating tool that we could potentially leverage and already learn a lot in the healthy populations that have been scanned in Human Connectome Project. So again, my long-term vision as we're developing these tools are to maximize the quantification, really look at physiology dynamically in the brain, but how it links to the body, and to be able to serve a population both for aging and dementia, but many other disorders. And in my acknowledgements, as I want to express my gratitude, I hope I've conveyed that it's really the interdisciplinary aspect of my training that led me to where I am now in the projects I'm in and what I'm also trying to build in my lab. One example of this is actually beyond the Neuroscience Institute. Miriam actually led myself and two of my cohorts to teach a mini course, which I've taken a lot of these sort of interdisciplinary strategies and integration of methods as I'm building my own BME courses at Davis. And I also just want to acknowledge that the network and the people that you meet are so critical. Oh, sorry. Here is me and my cohort mate, Rupa, who's a faculty member at UCSF, and our sons earlier this year. So I feel like as someone in Mamademia now, it's just great. It's great to be able to, have this interdisciplinary integration with life too. So I want to thank all of you for your attention, for the opportunity to speak, and all of my collaborators. Thank you.
Questions from Audrey, preferably from a postdoc.
Fascinating talk. Just a quick question about the leading and trailing part.
Yeah.
Was that solely determined by the estimated phase? But if the signals are periodic, how do we determine which one is leading, which one is trailing, just purely based on phase offset?
Yeah, so that's a great question, because if we were looking at bold signal from two different brain regions, then you'd have a clear sense of the timing, right? So in this case, it's not as periodic because we're not looking at breath to breath or beat to beat. It's the variability over the scan time. It still does leave the question of how to synchronize between heart rate signals and the brain, which are collected separately. And so we did a careful analysis of making sure that the expected global lags are consistent with the literature before we then which we accounted for, and then we were able to look at the vectors, if that makes sense. Thanks.
Hi, thanks very much for an excellent talk. I was curious, like in the last couple of years, there have been a handful of studies looking at how CSF flow in fMRI sequences. I was curious, like in the spirit of capturing all this information from dynamic imaging, if you've thought about how CSF might offer like complementary information, or if you think it's like redundant to the things you're already looking at.
No, I think that's an amazing question, because I think there are lots of studies that are looking at BOLD in CSF and correlating with these peripheral measures too. I would be really interested in both CSF specifically, but also perivascular signal, if there's a way to kind of get around the spatial resolution effects. I think that contribution and pulsatility certainly comes into play, and we haven't looked deeply into that. But it's a great question and really relevant, especially as CSF and diurnal variations change with aging, with dementia. So it's a really great question. Yeah.
Great talk, Audrey. It's so exciting to hear what you've been doing since you left Stanford. And
Nice to see you.
it's really interesting how you can separate these two phases, the early phase that leads and then the later phase that follows what might be representative of a heart rate kind of rhythm. And I was wondering, do you think that that first phase represents like luxury perfusion, and the second phase that leads more represents like systolic versus diastolic flow? And if that's true, could you deconvolute the signal and then get a more accurate map of where is luxury perfusion happening, and therefore where are neurons being activated?
Yeah, it's a really great question, and I don't think we have the temporal resolution yet, for instance, to dissociate like different components of the BOLD. But in bridging the two projects, I mean, if we continue to push the vascular fingerprinting approach, where we're actually more specific to quantification, then I think we can get at the biological mechanisms. At this point, we're considering the BOLD as a holistic signal, and I think that is relevant to resting state. But as you pointed out, biologically, it'd be really interesting to understand deeper what are the mechanisms. But thank you for the suggestion.
Thank you for the talk. Thank you for the questions. Let's give Audrey another round of applause.